was included on the UCSC like webinar list. So we might have non-ClinGen biocurators attending this call this time, but just to be transparent about that. And we also will allow them to share the YouTube recording after the fact. And I know that they're very interested in UCSC's use for BSEPs. So if you would love to provide them any feedback as they go along, or if they're soliciting feedback for that, I'll also offer that up. And then the next meeting for April 10th will be for the All of Us research program, both the Research Hub and the Data Browser. So we're excited for that one. And with that, I will turn it over to the UCSC team.
So thank you, Marina. I think from the UCSC, well, I am actually no longer at the UCSC, but I've been participating in what the UCSC wants to present here, so I will be presenting that. Well, my name is Anna Benet. As I said, I was 4 years ago, I was working for 2 years. I'm a clinical specialist. I'm a molecular biologist by training, and I lead the bioinformatics team at a diagnostics lab in Germany. And a couple of years ago, I went to the UCSC to work for 2 years, and just to think about how can the UCSC can be used for variant interpretation. So, or to assess a faster way of accessing data for variant interpretation. And what I would like to present here, it is well, it is a work that I've been doing with some of my colleagues at UCSC, and they will present themselves right now. But I just to introduce it is that in my lab, a colleague of mine, he's part of the BSEP, of the ClinGen BSEP for the HNPCC genes, so the Lynch syndrome BSEP. And we were always chatting and thinking about how can we combine tracks in the UCSC Genome Browser, because I guess every one of you have been at some point of their life, the UCSC Genome Browser interface. So how can we combine data tracks in a way that they are fixed and they are following they are representing the data sets that are recommended by the ACMG guidelines, or even more, how can the ClinGen curators use this genome browser to put their curated data in the form of tracks. And this is what I will present in a few minutes. But before, I think I would like that Lou Nassar, has been the engineer at the UCSC that has been creating this combination of tracks that I will show you right now. Lou, are you there? Yes.
Yes, I'm here. Hi. Yeah, I'm Lou. I'm one of the workers at the Genome Browser. I supervise the QA team, and I bring in data and tracks, and I've been working alongside Anna to put this stuff together.
And Max Haeussler, I'm not sure if Max arrived because he told me that he would come a little bit later. He's the principal investigator. He's the PI of the UCSC Genome Group. And he's also interested in, if you have questions, in chat with you, and in how do you work, how the different BSEP teams work, and to figure out if maybe, I don't know, maybe the UCSC, it's something useful that you can create your own tracks. But first, I think I will show you what we have done, and I think that then it will be everything much clearer for all of you. Okay, so I will share my screen now, and if you know a little bit about the Genome Browser, you know that there is a window where you can search for genes or positions, and then you have different types of tracks here. So in the middle of the screen is always the name of the track, and the track is showing data from any data set. Usually the data and the data sets are grouped here in these groups, and you can activate it or hide it. And we know that it is very overwhelming, all this data that you can find here. And what I did with my colleagues at my lab, we thought, okay, so why don't we put only together in a session the tracks that are recommended by the ACMG guidelines, the general ACMG guidelines, to find data, let's say lines of evidence in order to apply the ACMG codes. And this is what we did. So 2 years ago, we start developing, or Lou developed this together with other colleagues, what we call it the recommended track sets. And the recommended track sets are, one can find them here. If you click here, it's opening a small window. And a small window, it shows different tracks that they have been grouped together by specialists in data interpretation, and they think that they are useful for either interpreting clinical SNVs or CNVs. And now we are developing the non-coding SNVs dataset data. We have to update this, and also some other things like finding problematic regions in the genome. But what I will show you now, just to make a big overview, a wide overview on that, it is the SNVs, recommended track sets. And so when you click here, you land in this page that I have already opened. And here from up to down, we see the positions on the genome. We are now in the AZG gene, here in the AZG gene, and we are just in one of the positions. Doesn't matter which position it is. And I just want to show you the logical way of going through these tracks. So here there are some tracks giving information about gene disease associations like OMIM, gene reviews. Here there are the gene CC or the ClinGen gene disease validity classifications. This track here, it's empty because there is no validity classification for this gene, but in case we would have another one, we would have it here. The different colors and fields here, if you know the Genome Browser, it's just data from these databases. So here, for example, if we look at the GeneCC track set, you see all the entries that GeneCC has from this gene, right? So and in the mouse over, it's showing the data associated to this entry here. Like, for example, here, this gene is associated to microphthalmia, and it has supporting information. If one click here, you just automatically will open exactly here the details page for this item. So this is the link to the GeneCC gene page, and here are just informations from the GeneCC page. The format is not so nice and color as the GeneCC, but the information, it's the same one. If we go farther down here, there are the ClinVar variants, informations. Here are all the interpretations of the ClinVar variants. So if we click here, we will find all the submitted interpretations. I will not click everywhere because this will take very long, and I don't want to expand very long, just to show you the data sets. So here it is for the human gene mutation, and here the LOVD. Here we have the data from the mastermind variants. This is this company that it has an AI search machine, you know. So, and here the GNOMAD constraint metrics, the GNOMAD expression PXT, here the GNOMAD population data. Here some conservation data, and here the REBEL and the CADD variant pathogenicity prediction scores for missense variants. So this is just a very brief overview how if you have a variant and you can type here just the HGBS nomenclature of the variant, you will land at the variant position, and then you can just scroll down and see different data associated to this variant, and you can save sessions with the tracks that you need in your everyday life. And here we have this one. These are very broad, very global, just to cover the main ACMG recommendations, but one can design this for their own needs. And this is what I will show now. How we designed at our company these tracks for our own needs for the classification of BRCA1 and BRCA2 variants. So the Enigma Consortia published last August, I think the newest specifications for BRCA2 and BRCA1 and BRCA2 variants. And these specifications are very focused in special codes, not in all the codes, in very specific codes. And these informations and specifications are gathered here in the ClinGen specification registry. Here in this page. And what we just did, I will explain it in a very easy way. We just downloaded the tables from the specification criteria. So if we go to the Enigma specification criteria in the ClinGen registry, and here down on the page, you guys all all this, we have the different tables that the Enigma curators have curated their variants. So we just downloaded that, and we have tables like this one with a huge list of variants and curations. But for working in a clinical laboratory, the data that we need, it's not all the data related in these tables. They are very huge sometimes, but we need the variant positions and the codes and the code weight. This is the minimal information that we need to apply for the variant interpretation if we have these variants. And then, sorry, is that a question? No, okay. And then, so what we did is just we downloaded all these different type of tables and we put them in tracks. So this is the work that Lou did here. And I will show you now how these tracks look like. So now I'm again in a kind of recommended track set in another session of the Genome Browser. And here what we did, we put together all the data from these different Enigma tables. So for example, this track here, you see it in the middle. Sorry, the mouse over it's a little bit annoying, but this track here in the middle, it is the data from the table, Excel table that it's called Review Functional Assays for PS3 and VP3. I think this is exactly this table here. So we just took all these positions, all these variants, and we added these codes, and we have just mapped it against the genome, the human genome. And this is the result. So these are all the variants of these tables, and we colored, we put some colors, so green for more benign events, red for more pathogenic events, and black for the variants that they still don't have an assigned code. And in the mouse over, you can see the data from the Excel table, like the assigned code and the code weight. And if you click, for example, at this variant, it opens again the variant information from the table with all the other fields included in the table. So the other fields are not shown in the mouse over, but you can find them here. And another example, so the same, these are all the other tables. I don't think I will go through all the tables, but one table that I want to show that it was very complex for us, and it takes a lot of time to the variant interpreters at our lab to go through this table. It's this one. This is a table from I don't know if you know this table, but this is it seems that the Enigma variant curation group, maybe they are here in this call. I don't know it. I would be happy if they are here, if they have some comments on that. So they just put all the exon information to give codes, to assess codes to the variants that can cause splicing. So to the variants nearby the splicing, the exon intron borders. And what we did also, we just, or Lou transformed all this data in a list of variants. So we went through all these positions here. This is the beginning of the exon. This is the end of the exon. And this is the intron part, the intron variants. These are the positions here. You see here -1 and -2, and these are the last position of the exon in this case. And we applied also the codes that are given in this table. The codes are here, so it was a little bit complex to transform this data, but the data is very, very well represented and very well understandable in this table. And at the end, what we had, it was also another track. This is this track here. You see here, it's called variant codes for the splicing decision trees. And this track looks a little bit different because we had in this table the representation of single nucleotide variants at the, you see here, at the -1 positions in the well, +1 because it is on the other strand of in the reverse. So +1 position here. And again, this variant here could be assessed with a PBS1 code. In brackets, you see on the mouse over that it's RNA, because there is information also in these tables about if there are existing RNA functional assays on this variant or not. And here, this table has also the information of not only single nucleotide variants, but also duplications and deletions. And this is what we represented here. You see in the mouse over that this is the information for a duplication of the exon 19 in tandem. And here it is a non-tandem duplication, and how the ACMG codes are, in this case, different ones, or the strength associated to the code. In this case, it's a strong, and in this case, it's this moderate. And here the same for the deletion of the exon. So exactly, this is here we have another one where from another table that I'm not going to show you the table now. It's one of these tables from the specifications where they have defined the clinically relevant protein domains in the VRCA1 and VRCA2 genes, and we have translated also this and mapped it against the genome browser. So here you can very easily know in which domain and if this domain it is relevant or not. So we have added also other tracks to this session once we have the DIN, which we think we need them in our everyday life. And this is the ClinVar variants. It's also here. We have the HGMD and the LOVD. We have also added the BRCA Exchange Hub that Melissa Klein, I think she's here in the call, also designed together with Max Haeussler some time ago. And then here we have the GNOMAD data set. And what we have done here, the Enigma specifications, they recommend to use only the non-cancer controls, and we have set a filter to show here really only the variants of the non-cancer controls, not all the GNOMAD population, but only the ones that the Enigma specifications were recommending. And here, interesting, it is also maybe just exactly here Bias-Del. Enigma is recommending using the Bias-Del scores and not the Rebel scores, so we have changed the Bias-Del instead of the Rebel scores. So this is just to show you what we did with this data, and the people in our lab, it's a group of 35 people doing variant interpretation from exome and genome data, and they are very, very happy about that, I would say, because it has speed very much. They are much faster in gathering the data from all these Excel tables and do not have to consult all the tables. And so since we did this, we thought we could share it with the ClinGen group. Maybe they like this idea. Maybe this is things that they want to do. If you think that you would like to do, I'm sure that the Genome Browser people would help you to do this. this could be just private sessions for you to work, or if you like to open it to the wall, could open it to as public sessions, as, I mean, any possibilities that the ClinGen people thinks that it's important. And if there is one question, it's what happens when these tables change, because these tables are version, and I can imagine that the curation of these variants and of the genes, it changes, and the information in these tables is changes. We have not created now any automated process to update the data automatically, but I'm sure that the UCSC engineers have possibilities to work maybe with Excel tables that can be, if the format is always the same, can be updated or not, and maybe for the ClinGen groups to work together, or as I said, or to open the results to the wall. And I will stop here sharing my my screen. I hope this has been understandable, or I explain it well. I'm not an curation expert, but if you have any questions, or if you want to chat about that, or with Lou, wherever, or make your thoughts and come back to us, wherever. Thank you.
Thanks for giving that demo, Anna. It's really kind of identifies the power and the flexibility of the UCSC Genome Browser. So it's great that there's such a flexibility to work with groups, and also hopefully with expert panels, and how the plans to use those. The one question that Jessica Hatton put in the chat that maybe I can turn to you, Lou, if you're familiar with it, is if you have any knowledge of like the Rebel scores track, or Rebel scores in UCSC, and if you are able to display like the Rebel scores across multiple transcripts, or if you only display one right now, if you could give a little bit more clarity on that.
I'd have to look into it myself. I didn't work on the Rebel track, so it's not data that I put together or have worked more so than just knowing what it's supposed to represent. So if you want to leave an email, unless Anna has an answer, then I can look into this and then get back to you.
Okay, we can maybe follow up via email for that. So I think maybe if we could, for those who are on VSEPs or GSEPs, are there any questions about UCSC in general, or even thoughts of potential track ideas that you would find helpful for you, that we could have like a little bit more of an active discussion? I didn't realize we would have this much time left at the end of the call, so love to open it up to some discussion. And Deb, I know you're very involved in cancer expert panels, so please.
Thank you. Yeah, this was fantastic. And sorry, I'm not having my video on. I actually have a huge migraine right now. So I am so excited to see this work. So I do want to say, for anyone on the call that isn't, hasn't really dug into the UCSC Genome Browser, I always tell students that there is a there's a time investment to being an expert user on any platform, and you have to kind of choose which platforms are you going to do that with. I just have to say UCSC is one of them to do it with. It is an amazing resource, and the applications of this for the expert panels, especially seeing how you can align it now with the actual evidence codes, is, I mean, I think there's so many possibilities to it. So my question here is two. One is, are we able to search by that tag? So for instance, that evidence code, if we were to put in BRCA or put in a gene with that evidence code, would it pull us to, like, let's say a variant, or if we put in, how could we like kind of practically use it in the browser to search? Is that a possibility?
We'll have to, well, you will, Lou, have you understood the question? Do you want to answer, or should I answer? You are the expert on the tracks.
You can take a shot first, Anna, and then I can add on.
Okay, so yeah, so for example, in the browser, I think I feel comfortable if I have this open. But so for sure here you can look for the variant, but you will have to look for the variant. If you look for the gene, you end up on the gene view, and it is very, very overwhelming because there is a lot of data in these tracks. So first of all, the the visualization, it will be not very good. So the best thing, it is always to look for the variant and to look for the, you can type any HGBS nomenclature. And so it will direct you to the variant position. And then, of course, if you want to know the code, we can only display here the codes that they are in tables, or they have been assigned, the variants have been assigned. And then just looking for a gene and a code, you won't get it, the information, you won't get the variant that is associated to that code. I don't know if this was the question, but if
yeah, it was kind of trying to think of it in terms of like, how do we do this for other genes and other VSEPs, and what is the format of data that you would need? So like, I mean, I think to get this in the track in the first place, understanding how that was done would be really helpful on the ClinGen side, in order to get other genes into UCSC
at this level, you mean, or the genes at this level? Yeah, I think I don't know, how do you work? I guess are you working with Excel tables or with a kind of software, or
Okay.
yeah,
the
so
idea is to
everything's in the
Thank you.
so
I was gonna walk over on this ClinGen call.
sorry, Jeannie, I think you need to mute.
Yeah, we have someone unmuted.
Great, thank you. So we have it through the eRepo, the evidence repository, which does have an API. So we would be able to get all of the variants that have been curated and have these evidence codes associated to UCSC, so that they could be uploaded. So I think that I think we could get this for every gene across across that, that's in the eRepo, basically. But anyway, it's probably something we should follow up on. The possibilities are just great having seen this. So yeah.
Yes, Lou, do you want to say something, because you are an expert with the APIs and these things?
Yeah, that sounds reasonable. I think that's definitely possible, and we could do it. It's just about spending a little bit of time to find out how to create that interface to translate it into usable data. But we do this with other databases, and that it's a matter of following up, Deborah, like you're saying.
Great.
Yeah.
All right.
Regarding the searching, Anna covered it pretty well. So if you want to search by assigned codes, it's a little ambiguous. We could do that, but searching for specific variants is going to probably be the best. We can filter specific tracks so that if you're only interested in certain assigned codes, that can display, and that's something, again, we can also follow up on.
Great.
Now I see that Max is there. He arrived a little later, but now he's here. Hello, Max.
Hi, I just wanted to excuse, there was a huge traffic event here that completely killed everything. The first time I didn't have my computer with me. Sorry, I hope it went well anyways, but I'm here now.
Thank you. I just have also another comment on that, is that of course, if you have already a system that it's everything automated, then it's easy to load into the Genome Browser. The Genome Browser people can assess you this, but there is also, of course, the possibility to train people that you can create your own data tracks and your data hubs. You don't have to use exactly the same Genome Browser tools that they are using it, but you can create your own hubs. And if you have some engineers or some people that they are handy with this type of data, it is not very, very difficult. Or Max and Lou, we had also this, there is also this possibility that not the Genome Browser is doing all these things, but if the ClinGen group have people trained on that, can do it on their own, and then probably be much faster and be more independent of your work, Lou and Max.
Yeah, it's a great point. Definitely in our clinical lab, we've made our own like login sessions, like for example, for CNV analysis, where we load a certain number of tracks and they're just ready to go. So it's something we could even think about, like having just like a ClinGen representation of like the appropriate tracks, or even VSEPs could go as far as, having their own tracks and potentially like loading in some visualization of MAVE data, like you've kind of done with the VSEP functional data there. So definitely, as Deb said, the possibilities are endless here. It seems like it's very flexible as long as you have some creative thinking about displays.
And
Does
the
anyone-
data that Anna was showing, that is actually a track hub. So that's completely custom. So that visualization could be made by anybody and displayed that same way. That just has nothing internal to us. So it obviously, if we're more experts, we know how to make it look exactly how we want to, but that's something we can always work with people to, if they just tell us what they want to do with it, we noticed in some of your tracks looks a certain way, we want to display our variants this way, then we can always help and help you tailor it to look how you want it to.
Great. Do others have any questions or comments or ideas?
I mean, I think it'd be great if since we have a little bit of time and I'm a bit of an opportunist here, if we could go through and just show some of the tools that are available on the UCSC Genome Browser. I mean, just kind of maybe highlighting some of the tools that could be used for some of the work that our ClinGen curators do. And maybe even some of the tracks that are maybe the most important, I think, for curation, variant curation. So you showed that list of the tracks that are underneath the browser, and then all of the different kind of tabs that you can go through. But are there any from your perspective or insight that might be more beneficial in terms of curation, or does anyone else on the call use specific tracks that they might want to highlight as well?
Okay, sorry. Yes, I can start, and then if someone else can add something. I showed you here that when you go here under Genome Browser and recommended track sets, you open here the clinical SMVs and the clinical CNVs. And I showed you the clinical SMVs are just gonna, I don't know, there are just tracks. You can read them here. So I think these are the ones that we found in our lab, the most important ones for the curation of clinical small variants. And if you click at the clinical CNVs, these are the ones we found more relevant for the classification of CNVs based on the ClinGen CNV variant interpretation paper from, I don't know, I think it was 2020 or 2021. But here down, there is this part here, phenotype and literature. And in my opinion, most of the tracks here are the ones that are important for curation, because these are really the ones associated to clinical data. So here you have all the ClinGen, different ClinGen tracks. This is something we did time ago together with.
Yeah, I think that's a lot of the dosage.
Yeah,
one.
here's the dosage one. Now we have also the I don't know, sometimes it's a little bit difficult to find it. Here under this haploinsufficiency, these are the decipher haploinsufficiency, also the dosage things. And we have here some scores. Now we are adding many new prediction scores. Oh, and a very important one that it will be soon released is Splice AI.
Ooh.
So Splice AI, it's still not there, but it will appear here. I think it's it will be released soon. And here under this prediction score, always if you click here, then I will make now that it also open a window, then you can see here we have BiasDel, and we will add other ones. Now we only have BiasDel, but we are planning to add other ones. And it's also up to, if you want a special data set there, and this data set it's available, it's open source, we will just put it there. I mean, it's important to know what the users need, right? And here you have also the orphanate data, panel app data.
That's fantastic about Splice AI. So one of them,
Yeah.
I mean, a major thing at the field in general needs is ready, quick access to multiple Splice predictors. So I mean, Splice AI is one. Some others that are, I think, used at least on the ClinGen side are VarSeq is another one. But again, I don't know how publicly accessible a lot of those are not in terms of are you able to go to an interface and use it, but in terms of getting that into the format needed to pull it into UCSC Genome to be able to display it across multiple genes. So
yeah, we can check that. VarSeq, I know it. Yeah, I know the data set, but I don't know if it's available or not. But we can check on that, of course.
Great. And I also just want to mention again, sorry, I'm such a fan of UCSC Genome. So I just want to mention too, for biocurators on the call, those little signs below there that says pack and full, etc., those change how you view this data. And that can be very helpful because sometimes if it's in the full view mode, it's just too much information that creates a bit of the overwhelm and the vertical distance on the screen. Versus if you collapse it, you can get a sense of where the data you're looking for is, but it's not this long, vertical distance across your screen. So make sure, they've kind of preset which ones they think you want to have in the pack versus full, but you have the ability to change those and it can really make a difference. And then it saves kind of your last view or the view that you like. So you can kind of customize that so that each time you come back to it, you're viewing it with the tracks you think are important expanded and the tracks you want to know about, but kind of don't need to have there as well. Again, we have some biocurators on the call that are, a bit newer or maybe have not invested in using UCSC Genome. So.
Yeah, so what you were telling now, I will just show, for example, this track here. So in the middle of the window, so I will show you a couple of tricks maybe. So if you click at resize, I use very often this resize button. If you click at the resize button will adapt the window, the UCSC window to your screen. So I was before in a bigger screen. This is why it looked so big. And now you see here that this is exactly the window. So when you have, for example, this track here, if you move here, everything that it's in green, it's showing the track, what it's one data set. And in the middle, it's always the name of the data set. So here it is AGMD or here it is LOVD. Here, for example, this one, it's very, very huge. The amount of data, it's very big. And we think that if you click with the right mouse on the gray bar here, on the blue bar here, you can change the format of the track, exactly what you explained right now. And I want just to make a demonstration. So this is in pack mode. Pack mode, it's always the nicer one. But if there is a lot of data, it might be still overwhelming. I do not recommend to use full mode. I definitely do not. But if this is still too much, you can bring it to squish or dense. So I will just put it squish now, and then we will see what's happening. So you see now here, a lot of the informations are not there anymore, but the variant position is still there, and the information on the mouse over, because we always try to put the most relevant information on the mouse over, so that you don't have to click anywhere else to get a first idea of what is this variant, what is the information there. So the mouse over, it's still showing on. And for in each of the tracks, so we call them items. So every small item here, if you click, you will get more information on that. For example, if we click here now in LOVD, or let's click a ClinVar variant, because everyone knows very well what's behind ClinVar. So I just click to this variant, and here now it opens the information of this variant in ClinVar. And here you can just go to ClinVar, or usually here on the top of the table, there is always the link to ClinVar. And if I click here, I will just go directly to the ClinVar page. So if you prefer to read the data from here, and all the data that it's here, it is also in this page. So, okay. And for example, one very useful feature that I'm using is that sometimes biocurators, they have to work between HE19 and HE38. And sometimes you have a report that it's in HE19, an alt reported variant, but now you are working in HE38. And so you can jump very easily between the two assemblies. And this is done, so let's say I'm in this position, or I will just, let's say I'm analyzing now just this variant here, or this position here. Now I just zoomed in. See, now I'm zoomed in. So let's say I'm analyzing this variant, this position here, and I want to, now I'm in HE19. This is here indicated, and I want to go to HE38, the same position, but HE38. So I go to View, in Other Genomes, Convert. And then here I can choose to which assembly I want to go. And I want to go to HE38, make Submit. Again, here it's just telling you this is the new position, and it matches 100%. So this position, it's exactly the same. Just click here. And now I'm in the same position, but in HE38. And the good thing is that the same tracks are open. So I can continue curating from this new position in HE38. This is something that we need sometimes working with older data and then new data in the daily clinical lab. Yes, exactly. And then sometimes also something that I think it's useful. Sometimes one opens new tracks, so think, oh, let's see what is here, and let's open now also this track here. You can show this track here, and you make one makes refresh, and then the track format, it's changing. So if you want to go to the original tracks, you can always go to here to say reset all user settings, and then you start from 0. Because sometimes then the Genome Browser, if you are opening tracks and using other track sets, but then you won't go to a later point, you just make reset all user settings, and all these new tracks will just close again, and you will be at the beginning of your data interpretation position 0, let's say. Okay. Yeah, I wanted to show you this. Maybe this is useful.
Thank you, Anna. That was really helpful. I think a lot of those features are what curators would often use. Does anyone else have any questions?
Yeah, I see that in the sorry. Oh, sorry. I've seen that in the chat. This is about MAVE, the MAVE data, Max. We have been talking almost half an year about that now.
Yeah, and it's difficult for exactly the reasons mentioned in the chat. Some of the variants are not in genome. That throws off my thinking. There's another question in the chat about hotspots. I'm just looking at the website, and the hotspots are in. So if someone has PM1 hotspots, if someone has comments about that, how important that would be as a track. Yeah, I just want to throw that out because I'm just looking at the website, and I think there's no machine readable version of this. It seems like English text where you have from to amino acid.
Yeah, we've talked recently about structuring the hotspots where the VSEPs have defined the hotspots, enhancing the structure of that so that is computationally tractable from the CSpec, which is the criteria specification registry that Anna showed. So that is definitely an interest to try to structure some of the inputs that are given. I did wonder if I thought that cancer hotspots was available as a track, but I'm not sure that I see it right now. And then, of course, everyone hopefully should know about the protein domain track as well. We often pull up the protein domains, that can help with that specific evidence code. But yeah, hotspots itself, I don't see cancer hotspots on here. And the other was then getting that data into a format that we can send it to UCSC, or that UCSC could get it from the C-spec, which does have an API. It's just there is some processing needed on the input for that.
So the problem for me is with the hotspots, that's like an English text string that I can see. The couple examples I got by the chat, from the chat. Okay. Okay, but I have to look into, there are some comments now. So it looks as if it wasn't really structured, but like English text, but we can look into that later in the details. Okay, I'll look into hotspots.
Melissa Klein, I see you have a question.
Yes, hi. I want to speak to the question on MaveDB. I'm part of the MaveDB DCD workstream, and something that we've been working on is a genome browser track. I've actually got it on my to-do list to work on, hopefully today, more realistically tomorrow. But the DCD workstream's been mapping the Mave coordinates from whatever reference transcript the experiment was done on to a more standard reference transcript, and this browser track will do the next step of mapping those coordinates to genomic coordinates.
That's great. And Melissa, is that just for like certain, are you piloting with like certain Mave experiments? Like, is it maybe in the cancer space first, or like a large data set?
The initial data set that we're working with is pretty large. It's about 90 experiments.
Oh, awesome.
And in time, that's going to be hooked up directly to MaveDB so that we can update it automatically as new Maves come in.
Wonderful. Thank you for that work.
Thank you.
Yeah, so then the other question, and this is kind of what Deb had mentioned, but we had someone else put this in the chat, any thought of integrating cancer hotspot data and a link to cancer hotspots?
Yeah, I think we can look. I think Max mentioned, right, Max, that we can look at that. How is the format and to.
So for the hotspots, it seems that I got a confirmation from Christine Maloney that every group has a different way of writing that in the spec. So that will be hard to import.
Well, yeah, there's two sets of hotspots. So one is this cancerhotspots.org, and that's a tumor-based hotspots. And so there's two different things. The tumor-based hotspots from cancerhotspots.org is probably something that could be pulled in as a track. If it isn't already, I thought it was, but maybe it was in HG19, because the tracks can sometimes be different between the different things. So the second, though, is defining germline hotspots. And the germline hotspot definition is the one that is in C-spec. It is written in different ways. I'm always surprised at the number of different ways humans can think to write things. So the work that we'll be doing to structure some of the codes that we can structure now in a computationally tractable way, we will be focusing on those. So hotspot data is one. I think another one, of course, is like the BA1, BS1 cutoffs. We have a couple of ones that we're looking at to try and structure that in a way that we could get it into UCSC quite easily. But yeah, the separate question was on the cancer hotspots, which is just a completely separate data set from the expert panel definition of hotspots. Yeah.
The cancer hotspots are really easy to import. I agree. We can look into that. We have Cosmic, by the way. So
yes.
So that is kind of the same thing, I guess, but without an easy list.
Yeah, so this brings me to think about something. So as I understand this cancer hotspots from cancerhotspots.org. It is tumor data versus the other ones that you are talking about. It's germline data, I guess, right?
Yes.
And we were thinking also that we should separate we should not mix in one session tumor from germline data, but to have it always separated in different sessions or in different recommended track sets in the browser. I think you'd agree, right? Never mix tumor and germline.
So I think that having the idea of having a separate track set for tumor-based variants would be great, but having them available in the same session, I think would be really important, especially if you're looking at inherited cancers, you know, as well, and you want to look for like maybe double hits, one that, you know, has germline plus tumor. So having them allowing them in the same session would be great. Yeah.
Okay, I see. So we will make a ticket for the cancer hotspots.
But it sounds like we could follow up with UCSC on this in terms of making sure that we can get other genes, you know, expanding it from BRCA1 by using the evidence repository and the API to the evidence repository to pull in the evidence codes that are associated with variants into UCSC as a track. And then the second would be to communicate with UCSC when we have structured some of the material from the C-spec so that it can also be pulled into tracks. And so that's basically ongoing right now. And as soon as we're able to get that structured, then we can either hand it over to UCSC or maybe work with Melissa Klein on getting that in as well, given her background on creating those tracks too.
Great.
Yeah, I think that'd be great, Deb. And I feel like that falls really in the wheelhouse of your team, both with your interest and expertise in like the use of UCSC and also, you know, the Baylor team and like C-spec and eRepo and, you know, structuring that data. So hopefully, if you wouldn't mind like circling back around at some point or, you know, keeping this as an ongoing action item, that would be great. Okay, well, I think we are at about time. So I really want to thank again, Anna, Lou, and Max for, you know, this great discussion and demo and really just showing us the power of the UCSC resource. I think it has been great and we really enjoyed chatting with you. Thanks everyone for calling in and listening.
Thank you.
Thank
you
you.
for
Bye,
thank you
everyone.
for inviting us and organizing that. Thanks.
Yeah.
Bye-bye.
Bye.